All right, so today we're diving deep into Western blotting.
Oh, yeah, Western blotting.
I know, right? It sounds a little intimidating, but it's actually pretty cool once you get into it.
Yeah, and actually super useful.
We've got this awesome guide from Assay Genie.
Mm-hmm.
It's called their Western Blot Training, a comprehensive guide.
Okay.
And it's really interesting because it goes into detail on basically everything you need to know about Western blotting.
Yeah, it's like the perfect starting point if you want to learn how to do Western blots, or even if you just want to understand what all the fuss is about.
So think of a scientist in a lab trying to find this one specific protein,
Right.
like hidden in a sample with thousands of other proteins.
Yeah, it's like finding a needle in a haystack, but for proteins.
Exactly. And that's where Western blotting comes in.
Yeah, so Western blotting basically gives scientists this roadmap to follow so they can pinpoint that one specific protein they're looking for.
So how does it actually work? Can you walk me through the steps?
Sure, so it's a multi-step process.
Okay.
And it starts with sample preparation.
All right, so what happens there?
So you basically take your sample, which could be anything from cells to tissue, and you kind of break it open to release the proteins inside.
I see. So it's like almost like blending up a smoothie, but with cells instead of fruits and veggies.
Yeah, exactly. You're essentially making a protein smoothie.
Okay, protein smoothie. Got it.
And then the next step is gel electrophoresis.
Okay.
And that's where the real separation begins.
So what happens in gel electrophoresis?
So you take that protein smoothie and you load it onto a gel, which is like a jello-like substance,
Okay.
and you apply an electric current to it.
Oh, interesting.
Yeah, and because proteins have different charges, they'll move through the gel at different speeds based on their size.
So it's like a race where smaller proteins are faster
Okay.
and they zoom ahead while the larger ones kind of lag behind.
Yeah, exactly. You get these distinct bands on the gel, kind of like lanes on a racetrack, with each band representing proteins of a specific size.
Okay, I'm visualizing the protein race now.
And once you've separated the proteins by size,
Yep.
the next step is to transfer them from the gel to a membrane.
Okay, a membrane, why do they do that?
So the membrane is a bit sturdier than the gel
Right.
and allows us to actually visualize those separated proteins.
Oh, I see.
You can think of it like transferring a delicate drawing from a thin piece of paper onto a more robust canvas.
Oh, okay, that makes sense.
So it's like carefully preserving that separation we worked so hard to achieve.
So now we've got our proteins all neatly lined up on this membrane, but how do we actually find the one we're interested in?
That's where the antibodies come into play.
Okay, the protein detected.
Exactly. So we use these special antibodies that are designed to bind specifically to our target protein.
So it's like each antibody has its own unique protein target that it recognizes.
Right, they're very specific.
Okay.
So the primary antibody attaches to our protein of interest on the membrane. And then we use a secondary antibody that binds to that primary antibody.
Okay, why do they need two antibodies?
The secondary antibody is kind of like the signal amplifier. It's usually linked to an enzyme or a fluorescent molecule that allows us to actually see where our target protein is located on the membrane.
Oh, I see.
So it's like putting a little beacon on our protein of interest so we can track it down.
Okay, beacon, got it.
And the most common method for visualization is something called chemiluminescence.
Chemiluminescence.
Yeah, it sounds fancy, but it's basically a chemical reaction that produces light.
Oh, okay.
So when the secondary antibody binds to its target, it sets off this little light show that we can capture on film or with a special camera.
So we end up with a picture that shows us exactly where our protein is.
Exactly. It's like developing a photograph to reveal the hidden image.
That's so cool.
Yeah, and by analyzing the intensity of that light signal, we can actually figure out how much of our protein is present in the sample.
Wow. So not only can we see it, but we can also measure it.
Exactly. It's pretty powerful stuff.
So it sounds like Western blotting is a pretty smooth process.
Well, in theory, yes, but like any experiment, there are always challenges and things that can go wrong.
Right, of course. So what are some of those things that can trip scientists up?
Well, one common problem is poor transfer.
Okay, what does that mean?
So remember how we transfer the proteins from the gel to the membrane?
Yeah.
Sometimes those proteins don't fully move over, and you end up with faint bands or no bands at all on the membrane.
So it's like the protein transfer got lost in the mail.
Yeah, kinda. It can be due to a bunch of things, like using the wrong type of membrane or not optimizing the transfer setting.
Oh, I see.
It's like trying to fit a square peg into a round hole. You need the right tools and conditions for a smooth transfer.
Makes sense.
Another issue is nonspecific binding.
Okay.
So remember how those antibodies are supposed to be super specific to our target protein?
Yeah.
Well, sometimes it can get a little too friendly and bind to other proteins on the membrane as well.
Oh, no.
Yeah, and that creates all this background noise, making it hard to see our signal clearly.
So it's like trying to hear a whisper in a crowded room. It's just too much going on.
Yeah, exactly. To minimize that noise, we use blocking agents.
Blocking agents, what are those?
They basically bind to those non-specific sites on the membrane,
Mm-hmm.
kind of like putting up reserve signs so our antibodies only bind to our protein of interest.
Oh, that's clever.
Yeah, it's like clearing out the clutter so our signal can shine through.
Okay, so we've covered pore transfer, nonspecific binding, any other challenges?
Yeah, sometimes the signal from our protein can be just too weak to detect.
Oh, that's a problem.
It could be because there's just not enough of the protein in the sample.
Right.
Or maybe the antibodies aren't binding efficiently.
Okay.
It's like trying to hear a pin drop in a concert hall.
Yeah, tough to pick up that signal.
To boost the signal, there are a few things scientists can do, like using more sensitive antibodies or loading more protein onto the gel.
So it's like turning up the volume, got to make sure that signal is loud and clear. So it sounds like there's a lot of fine-tuning involved in getting Western blotting to work just right.
Yeah, it's a bit of an art as well as a science.
And that brings us to the topic of best practices.
Yeah, so there are a lot of things researchers can do to make sure their Western blots are reliable and consistent.
So let's dive into that. What are some of those key things scientists need to pay attention to for Western blot success?
Well, first and foremost, the quality of your reagents is super important, especially those antibodies.
Right.
Using high-quality antibodies is like having the right tool for the job.
Okay.
Low-quality antibodies can lead to all sorts of problems.
Yeah, like the non-specific binding we talked about.
Exactly. So it's worth investing in good antibodies to avoid headaches down the road.
Makes sense.
Another key aspect is optimization.
Okay.
Each protein is kind of unique,
Right.
and it might require slightly different conditions for optimal detection.
So it's not a one-size-fits-all approach.
Not really. You need to kind of tailor your protocol to the specific protein you're working with.
So it's like baking a cake.
Yeah.
You wouldn't use the same recipe for a chocolate cake and a cheesecake, right?
Exactly. You need to adjust the ingredients and the baking time based on what you're trying to make.
I like that analogy.
And lastly, reproducibility is super important.
Okay.
Western blotting can be a bit sensitive to small changes in technique or conditions,
Uh-huh.
so it's crucial to follow established protocols and use proper controls to ensure that your results are consistent and reliable.
So it's like following a recipe carefully so you get the same delicious cake every time.
Exactly. Consistency is key in science, just like in baking.
I'm starting to see the parallels.
And that brings us to the bigger picture, like why should we care about Western blotting?
Yeah. Okay, so we've got the basics down, but what does this all mean for us non-scientists? Why should we be interested in this lab technique?
Well, Western blotting might seem like this niche thing, but it actually has really broad applications.
Oh, okay.
It's used in research, diagnostics, drug development, you name it.
Wow. Okay, so let's break those down one by one.
Sure.
First up, research. How does Western blotting help scientists in their quest for knowledge?
Well, it's a fundamental tool for studying protein function and expression.
Okay.
For example, researchers might use Western blotting to see how protein levels change in response to a drug treatment,
Uh-huh.
or to figure out what role a specific protein plays in a disease.
So it's like having a powerful microscope that lets us zoom in and see how proteins are behaving under different conditions.
Exactly. And that knowledge is crucial for understanding all sorts of biological processes.
That makes sense. What about diagnostics? How does Western blotting help doctors diagnose and understand diseases?
So Western blotting can be used to detect specific proteins that are associated with certain diseases.
Oh.
Like, for example, it's used to detect HIV antibodies, to confirm Alzheimer's disease, and even diagnose some types of cancer.
Wow, so it's a really powerful tool for figuring out what's going on inside the body.
It is. It's like having a detective on the case looking for clues at a molecular level.
That's a great analogy.
And then in drug development, Western blotting is essential for evaluating the effectiveness of new treatments.
Oh, interesting.
Researchers can use it to see if a drug is actually hitting its intended target
Uh,
and to track how protein levels change in response to the treatment.
so it's like having a progress report on how the drug is performing.
Yeah, exactly.
Yeah.
It helps ensure that new therapies are safe and effective.
It sounds like Western blotting is a real workhorse in the world of science and medicine.
It really is. It's a versatile technique that has revolutionized our understanding of biology and continues to drive innovation in healthcare.
Well, that's pretty incredible.
It
I'm definitely starting to see why Western blotting is such a big deal.
Yeah, it's definitely a game changer.
We've covered a lot of ground today, from the basic steps of Western blotting to its wide-ranging applications.
Yeah.
And it's clear that this technique is playing a huge role in advancing our understanding of health and disease.
Absolutely.
Welcome back. All right, so we've got the Western blot basics down, but I'm curious, like what's next? Where is this technology headed?
Well, one really exciting area is the development of more sensitive and quantitative methods.
Okay, so like what does that mean?
Well, traditional Western blotting, it's great for detecting a protein,
Right.
but sometimes it can be a bit like trying to count all the stars in the night sky with just your eyes.
Okay.
You know they're there, but getting a precise count is tricky.
Yeah, I can see that.
So researchers are developing techniques that allow for more precise quantification.
So we're not just seeing if a protein is there, but actually how MUCH is there.
Exactly.
That's cool.
Yeah, and it's really important for understanding the subtle changes in protein expression,
Okay.
like in different biological processes.
Got it. So how are they actually doing that? Like what are some of these new methods?
So one really cool advancement is fluorescent Western blotting.
Okay, fluorescent so it glows.
Yeah, so instead of using chemiluminescence where a chemical reaction produces light, fluorescent Western blotting uses these fluorescent dyes that emit light at specific wavelengths when you hit them with a laser.
So it's like we're upgrading from a basic light bulb to like a high-tech LED that shines a very specific color.
Yeah, exactly. And the great thing about fluorescence is that the intensity of that signal, it directly correlates to the amount of protein.
Oh, that's handy.
Yeah, so you can actually measure how much protein is there just by how brightly it glows.
It's like having a dimmer switch on those protein beacons.
Exactly.
Cool. So fluorescent Western blotting, that's one big advancement.
Yeah.
What else is out there?
Well, another game changer is automation.
Oh, robots.
Pretty much. Imagine robots carefully carrying out those multi-step protocols we talked about, minimizing human error, boosting reproducibility.
So it's like having a team of tireless lab assistants working around the clock to make sure those results are consistent.
Exactly. And it's not just about efficiency, it's also about standardization, because by minimizing that variability, researchers can be more confident in their findings
Yeah, that makes sense.
and draw more meaningful conclusions from their data.
Okay, but with all this automation, high-tech stuff, I have to ask, is there any downside? Like, are we making things too complicated?
Well, that's a valid concern.
Yeah.
While these advancements offer tons of possibilities, they do require specialized expertise and sophisticated equipment, which can be a barrier for some researchers.
Especially those who maybe don't have as many resources.
Exactly.
It's like computers, you know? We've gone from those giant room-sized computers to sleek laptops and smartphones. But all that advanced technology, it requires specialized knowledge to actually build and operate.
That's a great analogy.
Yeah, so I guess finding ways to make these techniques more user-friendly and accessible, that's really important.
It is, absolutely.
So what about the future? Where do you see the most potential for Western blotting?
One really exciting area is the development of multiplex Western blotting.
Multiplex, okay, what's that?
So imagine being able to detect multiple proteins at the same time
Oh, wow.
on a single blot.
Okay, so instead of running separate experiments for each protein, we can kind of look at them all at once.
Yeah, it's like reading multiple chapters of the book at the same time instead of going through them one by one.
Okay, that would save so much time and effort.
It would.
And probably give researchers a much more complete picture of what's going on, right?
Exactly. It could totally revolutionize how we study protein interactions and networks.
Wow, multiplex Western blotting, that sounds like a game changer.
It is, for sure.
Anything else on the horizon that you're excited about?
Well, another promising area is the integration of Western blotting with other technologies.
Like what?
Things like mass spectrometry and next-generation sequencing.
Okay, so those technologies, they provide different pieces of the puzzle.
Yeah, they offer complementary information about the proteins,
Okay.
like their structure, their function, even modifications.
So by combining Western blotting with those other techniques, we can get a much deeper understanding of the proteins.
Exactly. It's about creating this powerful toolkit
Okay.
for studying these molecules.
So the future of Western blotting, it's looking pretty bright.
It is, yeah, both with challenges and opportunities.
All right, well, I'm excited to see where it goes from here.
Me too.
We've talked a lot about the technical side of things.
Yeah.
But I'm also curious about the impact.
Yeah.
Like, how has Western blotting actually shaped our understanding of human health and disease?
Well, it's been absolutely instrumental in advancing our understanding of countless diseases, from cancer to Alzheimer's to HIV.
Wow.
It's helped us identify those disease markers, understand how diseases progress, and develop new therapies.
Okay, can you give me an example of how it's made a difference in fighting a specific disease?
Sure, let's take cancer research.
Okay.
Western blotting has been crucial for identifying proteins that are overexpressed or mutated in cancer cells,
Yeah.
which has led to the development of targeted therapies that specifically attack those proteins.
So it's like having a guided missile that zeroes in on the cancer cells.
Exactly. And beyond identifying those targets, Western blotting is also used to monitor how effective cancer treatments are.
Oh, okay, so they can see if the treatment is actually working.
Right. By tracking protein levels over time, doctors can see if a treatment is working.
And adjust it if it's not.
Exactly.
So it's really a critical tool for personalized medicine.
It is, absolutely. And as our understanding of the human genome and proteome keeps growing, the role of Western blotting in personalized medicine is only going to get bigger.
That's really exciting.
It is.
I mean, it's amazing to think about the impact this technique has had,
Yeah,
both on healthcare and our understanding of human biology.
for sure.
Well, this has been a fascinating look at the world of Western blotting.
It has.
We've covered so much ground, from the basics to the cutting edge.
We have.
And it's clear that this technique is not only powerful, but constantly evolving. All right, so we've explored the ins and outs of Western blotting, but there's one more piece to this puzzle we need to talk about.
Yeah, the communication aspect.
Exactly. Like, how does Western blotting actually help researchers share their discoveries with the world?
Well, Western blotting, it's not just a lab technique.
Right.
It's also a communication tool.
Okay, so how does that work?
Well, it helps scientists present their findings, like they're telling a story, building a case, and convincing others of what they found.
So it's like a piece of evidence in a scientific courtroom.
Exactly. A well-presented Western blot with clear explanations, it can really make a scientific paper strong, especially when it's showing, the presence of a protein or sometimes even how much of it there is.
Okay, so imagine I'm reading a research paper, and I come across a Western blot. What should I be looking for to evaluate the data and see if it really backs up what the authors are saying?
All right, so first, check for those clear, well-defined bands we talked about before.
Okay, the protein race lanes.
Yeah, because a blurry or smeared band, that could mean there were some technical issues, like maybe the proteins didn't separate properly.
So it's like looking at a blurry photo kind of makes you question the quality of the whole thing.
Exactly.
Okay, what else?
Make sure they have the right controls.
The controls, right? Those are important.
Yeah, because they help make sure the results aren't due to some other random thing.
So it's like having a before picture to compare with the after picture so you can see the real difference, right?
Exactly.
Okay, so clear bands, proper controls, anything else?
Yeah, and then just step back and think about the big picture, Does the Western blot data actually match up with what the authors are concluding? Are there other ways to explain what they're seeing that they haven't considered?
So it's all about being a critical reader, not just taking everything at face value.
Right, like you're a juror in that scientific courtroom.
Yeah, weighing the evidence.
Exactly.
But, sometimes the world of scientific papers can be a little intimidating.
Oh, yeah, I get it.
Yeah. Even for someone like me who loves science, any tips for making it a bit less daunting?
Don't let the jargon scare you off.
Okay.
A lot of papers have introductions and discussion sections that are written in more everyday language.
Right.
To give some context, and focusing on those first can help you grasp the overall idea.
So start with the plain English translation before diving into the technical stuff.
Yeah, and then pay close attention to the figures and their captions.
Messages, right?
Yeah, they're like mini guides.
Okay.
That explain what you're looking at in that Western blot image.
So it's like having a museum guide right there with you whispering in your ear.
Yeah.
Pointing out the important details and helping you understand what you're seeing.
Exactly. And honestly, the more you learn about Western blotting, the easier it is to understand those papers and really appreciate the research.
And this has been an amazing journey,
It has.
From those basic Western blot steps to its cutting-edge uses and its role in scientific communication. It's been a wild ride.
For sure.
We've seen how researchers use this technique to unlock those protein secrets, diagnose diseases, develop new therapies.
Absolutely.
And we've even learned how to critically evaluate that evidence in scientific papers.
It's been a lot.
It has. But I feel like we've all leveled up our science skills today.
We have, for sure.
So to everyone listening out there, remember the world of science is filled with these awesome discoveries just waiting to be uncovered. So keep that curiosity burning bright.
Yeah, keep learning.
Who knows? Maybe you'll be the one making that next big breakthrough, Western Blot in hand.
You never know.
Ready to share your story with the world? That's it for our Western Blotting Deep Dive. Until next time, keep on exploring.